All right. Well, today we're going to talk about DNA replication. Um, when Watson and Crick and Franklin and others published the structure of DNA, one of the things in Crick's paper was that it had not escaped their notice that because the DNA is double stranded and there's complementary base pairing, that a possible copying mechanism was suggested immediately by the structure. So, what needed to be done was to demonstrate that, in fact, each of the two strands of DNA was, in fact, used as a template to synthesize a new complementary strand based on the complementary base pairing. So there's basically three hypotheses. The most obvious was what we would call the semi-conservative uh, model, where the two parent strands in blue here are separated and new daughter strands in red are made. If you have one round of replication, now you're going to have hybrid molecules that are red and blue. Second cycle, each of those strands are going to serve as parent strands, and then again you'll have with the blue one with a new red one, the red one with a new red one, and so on. Other hypotheses were out there. One was that the two parent strands somehow went back together. That wasn't very there wasn't really any mechanism to be suggested for that to happen, but it was a potential thing. And then the third, which was quite unlikely, was dispersive replication, and no one really believed that anything like this could happen. So Frank Stahl and Matthew Messelson set about doing an experiment where they were testing or tried to show that the semi-conservative replication process was happening. What they had at their disposal was an element, heavy isotope of nitrogen, nitrogen 15, which they could use to grow bacteria, and then the bacteria would have denser molecules of DNA. So we're going to call them D for dense. If you separate the DNA in a centrifuge tube, which we showed this before, the density of the DNA can be identified by staining it, and it glows orange. So they could watch in a test tube in a centrifuge what where the DNA went and measure its density. So if a conservative replication was happening, which is unlikely, you would start off with dense DNA, and after a round of replication in regular nitrogen 14, the new strand would be less dense, nitrogen 14, so we'll call it light and dense. You would have two bands. You would have the original density plus something that was lighter. In a semi-conservative replication, which is what people predicted would happen, you would have initially that dense DNA, but after one round of replication, you would have something that was less dense, but it would be a single density of DNA, which would be one strand being the light strand and one strand being the dense strand. After a second generation, you would have that same uh, heteroduplex of a light and dense DNA strands, but you would also have strands that were entirely made out of nitrogen 14 and be of lower density. So after one round, initially you would start out with one dense band of DNA. After one round of replication, you would have a less dense single band of DNA, single density. And then after two generations or two rounds of replication, you would have two species of DNA densities. With the dispersive, there would never be multiple bands. There would only be one band, and it would just change its density. So here's their actual data. You can see it at time 0. They have an initial density here. This represents the test tube. This would have been the top. This would have been the bottom. They were spinning this in a centrifuge. So dense is towards the bottom and light is towards the top. So you can see one single band after one replication cycle. You can see that there's still one single density molecule, but it's not as dense. And after two rounds of replication, you can see that there are two different density molecules. This one that is of that intermediate density, same as you had at one, and one that's a little bit lighter. And this continues on. And then down here, after they mixed this, they showed that mixing the original and the at the two rounds of replication together, you can see how all three of these, the most dense, the intermediate dense, and the lightest. So this matched the semi-conservative, which everyone thought would have been the case. So this was published in 1958, Messelson and Stahl, and so that supported then that the DNA was replicated in a semi-conservative manner. So one strand is going to serve as the template for a second strand. Working along at the same time on the enzymology of DNA replication, it was known that nucleotides would be added onto an existing 3' hydroxyl. So you needed a 3' hydroxyl in order to add a nucleotide, where the 3' hydroxyl on the sugar will interact with the phosphorus on the 5' carbon, and then another 3' hydroxyl would be available for the next step. So in terms of mechanism, people wanted to know, okay, did you start at a single position or origin of replication and synthesize DNA in one direction only, using one strand as the template, or did you synthesize in both directions, using both strands as the template simultaneously? So in other words, was DNA initiated and synthesized in both directions? Was it bidirectional or was it unidirectional? So they made use of radioisotopes again and did what was called a pulse chase experiment. So pulse chase. So you're going to expose what they were doing would they would have cells dividing in a culture on a petri dish. They would pulse them with a high radioactive dose of isotopes, and then they would chase that with by diluting out the radioisotopes. So you have a high radioisotopic labeling at the onset of DNA replication, followed by a trailing off of less radioisotopes as DNA synthesis proceeded. So what they observed were these sorts of structures. So this is one molecule of DNA, and you can see that there's a little spot where there's no isotopic labeling, and then there's isotopic labeling in high amounts, in approximately equal amounts on either side of that spot, and it trails off to lowering amounts of radioactivity. And then on the same strand, you'll have another position where it has started, and you have the same pattern, high to low, high to low. And a third spot, high to low. The lengths of these weren't always consistent, like this one is pretty long, these are pretty short, which implied that it perhaps started at this one first, started at this one second, maybe started at this one third. What you observed in each case was always an initial zone that would be the origin with high radioactivity on either side of that, trailing off to low radioactivity on either side of that. You didn't observe this type of pattern. This would have been the case if you started in one spot and went in only one direction from that initial origin. So this concluded that DNA synthesis was bidirectional from an origin of replication. The next question was, were both strands being simultaneously or nearly simultaneously used as templates? Were two new strands being made, or was only one new strand being made in each direction? So again, using a pulse chase type of experiment, except in this case, they gave it started with low and chased it to high radioisotopic things. So this is a electron microscope image of what they saw. Well, not an electron microscope. This is an autoradiograph. So you can see that there are two radioactively labeled molecules here, where you have less radioactivity here in the center area and more radioactivity out towards the edge. These molecules of DNA are the new daughter strands. The original strands, which I might draw in here in blue, the parent strands, were not radioactively labeled. So, what this experiment showed was that you proceed in both directions, but you also copy both template strands. Now, this posed a little bit of a problem because as you're making this new strand, you have to worry about the orientation of the DNA molecules. So, the bidirectional synthesis of both strands simultaneously can cause of synthesizing only in the 3' direction. So this would mean necessarily that if you started at one spot, you would have a strand that began and continued, but you would also have to have starts and stops on the other strand. The other strand would be called the lagging strand, would mean that you couldn't start here and go this direction. You have to go from the 5' to 3' direction, which would be this way. So that would there hypothetically there should be short fragments, or there should be some sort of difference between these two strands that one could identify. So a scientist by the name of Reiji Okazaki and his wife Taniko identified these, or isolated and found these fragments, and now they're called Okazaki fragments. They turned out to be about 1,000 to 2,000 nucleotides long. And interestingly, the beginning of each strand was not made from DNA, but was made from an RNA. And they called this a primer. It got it started, it primed it. And that little stretch was about 15 nucleotides long, right at that 5' end. So these Okazaki fragments could be isolated. Here they are showing you the size of these fragments with uh, the RNA, which was at about this is 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So this is about 14 base pairs, 15 base pairs of RNA that's at the end of that Okazaki fragment. So the question was, why is there an RNA molecule at the beginning of the DNA molecule? And how do these Okazaki fragments get turned into a single DNA strand? Because we know that the DNA strand does not exist as a bunch of little fragments. So we'll get to that as we talk about the enzymology of this process a little bit later. Those fragments do get joined together. So let's take a look at some of the various terminology that deals with replication. First off, the site where it starts is called the origin, or it's going to always be labeled ORI on the DNA. In bacteria and viral genomes, there's usually a single origin of replication. Some viral genomes consist of multiple chromosomes, or multiple strands. So you're going to have multiple origins with those. In eukaryotic cells, we always have lots of chromosomes, so we're going to have multiple origins of replication. We have thousands of them on each individual chromosome. So beginning at that one origin and expanding out over an area that gets replicated from that origin, we have a region of DNA that we call the replicon. So it is that region that is copied from that particular origin. So in the case of bacteria, the entire chromosome is the replicon. In the case of, say, a eukaryotic chromosome, each origin is responsible for several thousand, maybe 50,000 base pairs of DNA in a replicon. Okay, each replicon is going to have a fork, a replication fork on each end that are going to be moving in opposite directions. Okay, so we know that the DNA is bidirectional. You're going to start in the center spot and you're going to move out. Okay, the each fork is going to contain 4 DNA polymerase enzymes, or we call it a holoenzyme because it contains all the little parts, all the little machinery to replicate the DNA. So each fork will have 2 enzymes, and there are 2 forks, so 4 total enzymes working on a replisome. What does a replication origin look like? So there are specific sequences within the DNA that can be identified. In E. coli, the origin of replication is called the origin C for the E. coli chromosome. And this is the sequence. You have about 250 base pairs here, 245 base pairs, and there are 13 base pair sequences that are quite similar. They're not identical, but they're very similar. And they are AT rich. So you can see, right, lots of A's and T's. And then spaced down a little bit from that, you have 4 sequences that are 9 base pairs long that are also almost identical. And they are not AT rich per se. Okay, and each of these sequences are the same every time. So when you find DNA sequences like this that are conserved and repeated, you immediately think this serves an important purpose. Why is this DNA being conserved? Why are these sequences the same every time? And the reason is usually that promotes interaction with a particular protein. There's going to be some mechanical reason that this sequence of DNA is conserved like this. We see the same trend in yeast. Okay, so the origin, it's also known as the autonomous replicating sequence, has a similar type of arrangement where you have 3 sequences that are highly conserved, and then this 11 base pair sequence down here that is a little different. But these 3 boxes, B boxes they're called, and this 11 base pair sequence are all critical for DNA replication. You'll notice that this 11 base pair sequence is very AT rich. In fact, all of these are a little bit on the AT rich side of things. So, what goes on at this origin of replication? So, for these events in E. coli, where it's perhaps the best well understood system, we know that the 9-mer repeats are recognized by a protein called DNA. The DNA is bound by that protein, and it forms a complex. It's not just one protein, but there's a complex there. And the DNA is twisted by binding to that protein, and this induces conformational changes, which this sequence here can be unwound, where the 13-mer repeats are located, which are AT rich. And those strands of DNA, as they are separated, are held separate by specific binding to a protein called single strand binding protein, SSB. They've just lightly shaded it in here. Once this has opened, and probably what's actually driving its unwinding is the interaction of DNA C, DNA B proteins. So DNA B is in fact a helicase, and DNA C is an accessory protein that helps the helicase form a complex. So 6 subunits of this will interact with these 13 more repeat areas, and one group of them will be facing one direction on the top strand, for example. The other will face the other direction on the bottom strand. So these helicase proteins, helicase is an enzyme which will cause the unwinding of the DNA helix. So the helix is loaded on to the 13-mer repeat region after the 9-mer repeat region has been bound by DNA A, which contorts it, which makes it able to be melted, which makes DNA C able to load DNA B, helicase, onto that strand. Okay, once you have it opened up, now the strands will continue to unwind. Once they've unwound for a little distance, then the rest of the DNA replication machinery can enter. So an overall structure of a replicon can be shown here. You had an origin where you have the initial binding of DNA A and helicases. Once the helicase proceeds to unwind this strand, so one helicase is going to go this way, one helicase is going to go that way, and the helicase will be wrapped around the leading strand each time. The DNA A, which would have been here, is booted off out of the way, and you just have the helicase. Once the helicase is past a certain distance, then you have interaction of the rest of the DNA replication machinery. And that machinery is going to be located here nearer to the actual fork. So you're going to have a little bit of difference in the machinery on the leading strand versus the lagging strand. The lagging strand is going to need to intermittently restart synthesis after it has proceeded about 1,000 to 2,000 base pairs. Whereas the leading strand is going to just continuously synthesize new DNA. And again, on chromosomes like our eukaryotic chromosomes, there's going to be lots of origins of replication. Each origin is going to direct a replicon of maybe 50 to 100,000 base pairs. And the sizes are not uniform. They just go until they reach another replicon. So some of the proteins we've learned so far, their mutants were identified genetically. So that's why they have these names. These are mutations that affected DNA polymerization in E. coli, DNA A, DNA B, DNA G. So we saw DNA A is the initial binder to the origin. It recruits DNA B and DNA C, brings in DNA B, which is a helicase. The helicase is important that it unwinds the DNA ahead of the replication fork. This HU is another protein, and specifically in E. coli, this is more like the DNA C. DNA G is a primase, so we saw that the Okazaki fragments started with an RNA primer. It turns out the leading strand of DNA also starts with an RNA primer. So this enzyme called primase synthesizes those RNA primers. Now, in all of this, the DNA is a double helix. So as you unwind it in one area, it's going to want to wind more tightly in another area. And so that is called overwinding or supercoiling. And that has to be relieved every so often, or else it's just going to get tangled up. This is sometimes known as a topological problem in DNA synthesis. You have a conformation, a topology that is going to become a problem. The enzymes that can relieve that are known as topoisomerases. So they will cut the DNA strand and relax it. They will just cut one of the two strands, and it lets the other strand swivel around, and it will allow for a relaxing of that overwinding. DNA gyrase is the name of one of these enzymes. Some of them are just called topoisomerases 1, topoisomerase 2, and so on and so forth. You have type 1s and type 2s. The mechanism of how they actually cut the DNA is what gives them the type 1 or the type 2 name. Not really important. So what about the actual building of the DNA molecule? Okay, so a fellow by the name of Arthur Kornberg started looking at DNA polymerization in E. coli and tried to identify the enzymes involved. First enzyme they identified, well, they called it DNA polymerase 1. And this enzyme is a relatively small enzyme. It's a single subunit that had the ability to synthesize DNA from a 5' towards a 3' direction, but it also had some other activity. One activity was that it would actually back up. It would go from the 3' to the 5' direction, and it would cut out a nucleotide. So that's an exonuclease activity. But it would only do that if the nucleotide that was placed in was incorrect. So that's basically a proofreading function. If the wrong nucleotide comes in base pairs, say you've got a C here and you need to put a G in, what if an A comes in? It gets attached. Well, it's not going to base pair properly. The enzyme is sort of stalls. So the enzyme immediately cuts that one back off. That's proofreading. It also had a 5' to 3' exonuclease activity. And this is associated with its ability to remove the RNA primers, which is one of its primary jobs in real DNA polymerase activity in the E. coli. So this means you've got a strand of DNA and you've got another piece of DNA here, and you're coming along and you bump into this. Well, this needs to get out of the way. So this enzyme can degrade that as it makes new. So in reality, DNA polymerase 1's job in the replication of DNA is to remove the primers and fill the gaps left by the primers during DNA synthesis. It also fulfills a DNA repair mechanism in normal life of bacteria. Its job is not the primary DNA polymerase of E. coli, even though it was isolated first. Okay, so they went on to isolate other molecules, and they found a DNA polymerase 2 and then realized that wasn't the whole thing. They found DNA polymerase 3, and this is the complete enzyme. So it's a multiple subunit enzyme. It has subunits that are going to function as the 5' to the 3' polymerase subunits. They're building DNA, and it has subunits that work for proofreading. It will not have any 5' to 3' exonuclease activity. So this enzyme, DNA polymerase 3, okay, so a lot of times we just call it DNA POL 3, Roman numeral 3, is the primary DNA synthesis enzyme in E. coli. And we understand quite a bit of how it works. In eukaryotic cells, such as human cells, we have multiple DNA polymerases. The primer is known as polymerase alpha, and it synthesizes those little primers that we need, RNA primers. The lagging and leading strand, there are two separate enzymes that function in that respect, but they function almost identically. And then we have DNA polymerases that exist within mitochondria and chloroplast. So gamma is a mitochondrial DNA polymerase in humans. This is a cartoon of the protein structure for E. coli DNA polymerase 1. So all these enzymes have similar structure, okay? So if you look at this, you can see here is kind of the this is called a ribbon diagram. This is also sometimes known as a well, I don't know, dynamite diagram. Little sticks of dynamite or something, rods and ribbons. There's a cleft. That's the active site where the DNA strand that's being polymerized or copied is going to lay, and the new strand that's being made is going to have its 3' hydroxyl right in this cleft. Okay, so here's the enzyme. You can see the blue and the green here, and the red form kind of a pocket. Okay, and here too, there's kind of a pocket, and there's always a metal ion in there, like magnesium or something that's catalytically important. And so the polymerase kind of holds on to the one template strand and is synthesizing that second strand in that active site. Here's some other different DNA polymerases that are kind of the same. All right, so What are the different functional subunits? Well, there are two. They're set up in pairs. So there's two primary polymerase subunits. This is the alpha, these two dark green ones, and then there's going to be a pair of enzymes that are known as epsilon and theta that work together in the exonuclease proofreading. So this is synthesis and this is proofreading. And I don't know who drew this, but you know, this is not what the enzymes obviously really don't look like this. Both of these active subunits, okay, so the alpha, epsilon, theta subunit is the polymerase itself. That's listed up here right as the core, okay. They're going to be held together or tethered together by a protein known as the tau protein. This is probably a better representation of it here. So these core polymerases are held out on the ends of these tau proteins. And the tau protein interacts with a complex. It's a heteromeric complex. It's got several different proteins in it, known as the clamp loader. So the clamp loader is going to actually interact with tau, hold it together. It's going to also interact with the primase, not shown on here, and it's going to also interact with a protein called the sliding clamp, sometimes known as the beta subunit, the sliding clamp. All of this stuff together represents what we call the holoenzyme. The clamp, that sliding clamp, what's its job? Its job is to wrap around double-stranded DNA. So in E. coli, this clamp is made from two proteins. They come together kind of like either half of a donut and clamp around the DNA strand. Then they're attached to the clamp loader, which is over here. And they're going to be attached to the DNA polymerase as well. In mammals, we have three proteins. They're kind of like light tan, orangey, blue, but does the same exact thing. So instead of three molecule, or instead of just two halves, it's got three parts, but it wraps around the DNA. So these clamps allow the DNA polymerase to stay attached to the DNA for a long distance of synthesis. They make the DNA polymerase what we call highly processive, so that it processes a large stretch of DNA, synthesizing it without turning loose or without stopping. That's pretty important when you're trying to replicate 50,000 base pairs of DNA. You can't just start and stop. All right. So the sliding clamp holds DNA polymerase onto the DNA. All right. So remember, we have to add on to only the 3' hydroxyl. So for these DNA polymerases, they're called DNA-dependent DNA polymerases. They require a DNA template, right? That's the parent strand. They require a 3' hydroxyl. So that's why there's the requirement for having a primer. That's even made out of RNA. Okay. The deoxynucleotide is going to be added onto that growing 3' end. So we say that synthesis is 5' to 3'. Why an RNA primer? So the primer, the primase, would be called a DNA-dependent RNA polymerase. And it does not require a primer. The enzyme can hold the nucleotide and add the second nucleotide to it and so forth. So priming with RNA does not require a free 3' hydroxyl for the very first base. But to add DNA to something does. So DNA polymerases require a place to add the nucleotide, or else they won't start the polymerization process. So this diagram shows what's going on the lagging strand and talks a little bit about some of the other proteins that are involved. So there's going to be a DNA polymerase 3 replicating the leading strand. There's going to be a DNA polymerase 3 replicating the lagging strand. Okay, again, this is in E. coli. The thing that isn't being shown here is the tau proteins, which would tether these together, the clamp loading proteins, which would be loading on a clamp, and they're not really showing you the clamp itself. So they're missing some of these components. In order to show you that there's a primase, which just synthesized this RNA primer, and to show you that there are regions of DNA that are single-stranded, those are going to be bound by single-strand binding protein. There's the DNA helicase, which is going to be on one of the strands. There are topoisomerases, which are not necessarily located here, but can be located anywhere on the DNA molecule to relieve overwinding. After an Okazaki fragment has been synthesized, the primer's sequence, the RNA sequence, has to be removed. Okay, that's done by DNA polymerase 1. DNA polymerase 1 will recognize that there is a nick at the end of an Okazaki fragment where this fragment will come up and stop here. It will start at that nick and it will remove the primer. So this one shows that the primer has been removed. There should be a little red patch right there and it's gone now. It will remove that primer and in its place synthesize DNA. When it gets to the next strand, it will stop. Okay, there'll still be a gap there. There'll be where the two strands of DNA are not formed, do not have a phosphodiester bond between them. So to finish that formation of that phosphodiester bond requires yet another enzyme known as DNA ligase, which is shown here, this blue torus. So the DNA ligase will covalently link the Okazaki fragments. This shows both strands, shows the polymerases running opposite directions. Okay, so what we know is that they do not run opposite directions. We know that the entire fork goes one way. We know this because we saw the data that showed it was bidirectional and that both strands are being synthesized nearly simultaneously. So there's a little bit of contortion that has to go on. So the helicase will be unwinding a strand of DNA, and that strand will be looped out. The clamp, which would be here and here and here on these DNA polymerases, and the clamp loader and the tau proteins are all here, basically in the same area taken up by the primase, this green. Okay. So the primase will lay down a primer, and the DNA polymerase will, on the lagging strand, will be synthesizing until it hits the existing primer of a previous Okazaki fragment. When it does so, it will turn loose. It will come back and grab this primer and begin to synthesize this region here. Okay. In the meantime, everything has progressed further down, and another region from here to here, let's say, has been looped out and is single-stranded. To keep that single-stranded DNA protected and keep it from winding up, it's coated with single-stranded binding protein. So the lagging strand is going to be opened up into loops, and those loops are going to be replicated individually as Okazaki fragments. The leading strand just continues its replication continuously. There's a movie animation of this that is on D2L that you should open and watch. This shows, again, that same sort of sequence of events, but shows it as some sort of snapshots. So here you have everything shown in here, the tau proteins, the leading strand synthesis over here, the lagging strand synthesis over here. So notice that the 3' hydroxyl is here going this direction on both of these. So as this reaches the end, it's going to bump off single-stranded DNA binding protein, and it's going to come up till it gets to the end of the new primer, which is in gray. Okay. When it does so, it stops. When it stops, it releases the from the sliding clamp and comes back. So it stays attached to tau, but it releases the sliding clamp. Okay. Now it comes back and it's going to have a new sliding clamp added to it. Okay. And it's going to be bound over where the primase just laid down a new primer. So primase is uh, synthesizing that new primer, and the clamp is actually being placed around that primer because the clamp needs to go around double-stranded DNA. It could be double-stranded DNA or double-stranded DNA RNA, complex. It doesn't matter. So here it goes again. Now this will synthesize this entire little region right here that's covered with single-stranded binding protein. In the meantime, a new loop will form behind it. So again, there's a replication animation on D2L. You should go and watch. So DNA polymerase 1's role is to come in. It recognizes that there's a nick here, okay, this single base pair gap. So there's no phosphodiester bond right here. Okay, so that's known as a nick, N-I-C-K. DNA polymerase 1 will bind to that. Also, at that position is the sliding clamp, and DNA polymerase 1 is recruited by the sliding clamps. So it will then remove or excise the RNA nucleotides one at a time and replace them with a deoxynucleotide where it's attaching it to the existing 3' hydroxyl group. Okay, it'll keep doing that until it's removed all of the RNA. When it gets to the end of the RNA, it'll remove a couple of these nucleotides, but it won't go very far because as soon as it removes a deoxynucleotide monophosphate, that inhibits the activity of DNA polymerase 1. And so it will stop. Okay, so it will basically remove exactly the primer plus maybe a couple of bases, but it will not join that base to the next base. It will still leave a nick. Okay. And that nick is filled in or is joined by DNA ligase. On circular chromosomes, the DNA polymerase just needs to go around each strand, right? And the primers are removed. You filled in with DNA polymerase 1, and it's ligated. Okay. In reality, you create two chromosomes that are actually interconnected rings like this, okay, like magician's rings. And those need to be unconnected. That connection is called catenation, and they need to be decatenated. And that's done, again, by one of the topoisomerase enzymes. On linear chromosomes, DNA polymerase gets to the end of the strand, and there's a bit of a problem because it will not be able to fill in where the primer was at the very end of the chromosome. This region of our chromosomes, we call them the telomeres, have a repeated structure, which is going to help with keeping this end of the chromosome intact. So the telomeres have sequence TTAGGG for human and a lot of other species of telomeres. There are somewhere around 100 to 300,000 base pairs of repeats. So each repeat is 6 bases. That means there's something like 50,000 repeats of that 6 base pair sequence. This shows some of the telomeres in different organisms. You can see here's a fungus, has the same as us. So there's some plants, they have 7 instead of 6. Yeast has a weird one. Some of them have all G's and T's, and some have a C thrown in there. So these 6 to 7 bases, except for in case of yeast, repeats of sequences at the end of the chromosomes are going to serve to protect the chromosomes from degradation during DNA replication. So telomere repeats are shown here. And the very end of the telomeres, there's going to be a strand of single stranded DNA. It's about 300 nucleotides long. This is called a telomere overhang. We know that when DNA replication occurs, some of this overhang is lost every time. So you're going to have a reduction in the overall length of the telomeres with each round of replication. All right, so at these telomeres, we know that they get degraded each time DNA replication or rounds of DNA replication results. And that's because there's going to be a primer put on out here, right? But that primer is going to get removed, and that's going to mean that the strands are going to get shortened every time. So we think in mammalian somatic cells, there's somewhere around 100 to 1,000 telomere repeats, right? So 1,000 repeats, be 6,000 base pairs. With a total of, say, 300,000 there, and you lose 6 every time, you get about 50 replications before the telomeres are gone. That would mean that when the telomeres are shortened to that amount, the replication will not happen again. And we know this lines up pretty nicely with something known as the Hayflick number. The Hayflick number was identified by a fellow by the name of Hayflick, his last name, that cells, if you take cells from an organism, primary tissue culture cells, these are not tumor cells, they're not diseased cells, they're just normal cells like the fibroblast cells under the skin that regenerate our skin. And you put them in a tissue culture environment, they will divide, but they will only divide about 40 times, between 35 and 40 times. And they just will not divide anymore. And this sort of maximum number of cell divisions was known as the Hayflick number. And that correlates pretty nicely with the fact that we know that with each round of DNA replication, each time the cell divides, it had to replicate its DNA. So it lost telomere repeats. And of course, you're starting with cells that have already replicated at some point to some degree. So if you've already done that a few times, you're down to around 40 replications and you won't be able to replicate any longer. So it's thought that telomere length is sort of a way of timing the number of cell divisions that a cell can go through. And it sort of pictures in or plays a part in what we call cell senescence, where a cell just won't divide after a certain number of times. There are cells that need to maintain those telomeres, these cells like the cells for the gametes. Okay. The telomere sequences need to be the full length so that the next generation of organisms, starting with that gamete, has full length telomeres. We also know that during early embryogenesis, telomeres are extended. So this Hayflick number, this count, doesn't start right off the bat with the very first cell. So how are telomere sequences maintained? There's an enzyme known as telomerase. So this enzyme has, as part of its enzyme structure, a RNA template, an RNA molecule that it uses as a template to extend the 3' end of one of the telomere strands. The enzyme itself, telomerase, is what we call a reverse transcriptase. It is an RNA dependent DNA polymerase. So it uses this RNA template to synthesize a DNA strand. And making DNA from RNA is called reverse transcription. So the RNA that the telomerase has, its template, is complementary to the telomeric repeat that we see in our telomere sequences. So we can see here that you would have a telomere, and there would be a region here where it has lost the primer on this strand, and this strand would be single stranded, and it needs to be extended. So the telomerase enzyme has this short RNA molecule. A, a part of that molecule is the template part, not the whole thing, and it essentially can base pair with the last three nucleotides here, the of our telomere, and then serve as a template to extend this 3' hydroxyl out. Okay? So it extends it out. When it gets to there, it can skip down and extend again and again and again. Okay. This provides enough of a stretch of DNA here that new DNA synthesis can be made by traditional priming and synthesis. So the telomere length can be maintained to a degree. We know that in most cells, in humans, for example, most somatic cells in our body, telomerase is inactivated. The enzyme is no longer expressed, and telomeres definitely shorten. It's been noted that in many tumors and many cancers, that the telomerase gene has been reactivated, and that uh, reactivation seems to coincide with the ability of those cells to be what we call immortalized or transformed. They're transformed from cells that would be normally regulated by only being able to divide a certain number of times to essentially being immortal. They can divide forever, and they can form these cell lines that can be propagated in a tissue culture environment for forever. One example is the HeLa cell. HeLa cell was named from Henrietta Lacks, a woman who they took these cells from without really her having any knowledge that they were going to be used in the way that they've been used. But those cells were cultured and studied because they were from a tumor, from uh, is either ovarian tumor or a uterine tumor, and those cells were transformed, and they have telomerase activity in them. The end, there's still always going to be an area at the end of the telomere that's not base paired, and this forms a loop, or can be used to form a loop called the T loop at the telomere, where you have a region of the DNA that will open up, and this single stranded tail will be able to base pair. And you have what's known as a D loop or displacement loop. And this seems to be rather stable, and this is all bound by proteins. And this protects the end of the chromosome from undergoing any degradation. So, in most of our cells, single stranded DNA is something that the cell does not like to have in it, especially in the cytoplasm. Of course, this would not be in the cytoplasm, this would be in the nucleus, but during cell division, it can be exposed to the cytoplasm. And there are enzymes which degrade single stranded DNA, and this is in part to protect against things like viral infections. So, when it degrades the single stranded DNA, it's going to go a little bit too far, a little bit too far, and so forth. So, to protect that, the DNA is looped into this duplex. All right, well, we'll stop there with DNA replication, and we'll talk a little bit about some of the consequences of the enzymology of DNA replication and how we have used that to actually, as a tool, to either make DNA in vivo or to synthesize DNA so that we can sequence it. So, a fellow by the name of Fred Sanger uh, figured out how to sequence proteins, won a Nobel Prize for it. Later on, he figured out how you could use DNA polymerase to sequence DNA. and got another Nobel Prize for that. And this method of DNA sequencing has been around for quite a while now, and it's sometimes called Sanger sequencing, or it's more often known as chain termination or dideoxy sequencing. So it makes use of the fact that for DNA polymerase to add a nucleotide, there has to be an existing 3' hydroxyl. If you use a base that is missing that 3' hydroxyl, right? It's going to be missing the 2' because it's deoxy. Now it's missing the 3', so it's dideoxy. 2', 3', dideoxy nucleotide. So it can be added, but nothing can be added to it. So once this nucleotide is added to the growing chain, it terminates any further synthesis of DNA. If you could identify the base that was attached to this and was placed as the last position, then you could identify a particular spot in the DNA. So this led to the idea of chain termination sequencing. And that's really shown here. So if you had a template molecule of DNA, you would have millions of these templates, and you would divide them into 4 separate reactions. In one reaction, you would place dideoxy C, TP. In another reaction, you would place dideoxy G. In a third, dideoxy T. And in a fourth, perhaps dideoxy A. Okay, so you have 4 separate test tubes. Each test tube has many molecules of the template. You provide a primer that can base pair to that template, and then you add the dideoxynucleotide plus a certain amount of the regular nucleotide. So in this reaction, for example, it's going to contain dideoxy CTP, but it's also going to contain regular DCTP as well, plus DATP plus DTTP plus DGTP. So what that means is you're going to start synthesis, and sometimes you're going to come to the G here, and it's going to put in a dideoxy C, and that's it. So this strand will never get any longer. But it could start on a different template. It could have started and it could have put in a regular CTP here and continued on to the next. Or it could have put 2 regular ones in and continued on to the third or to the fourth or to the fifth. So if you have several millions of these templates, you can continue on through several millions of these, and you'll be stopping at a statistical number at each position where there's a C. This is going to create fragments of DNA of distinct lengths that are going to terminate with the same nucleotide every single time. Okay, same thing is going to be happening with the T's and the G's and the A's. So you're going to create a, a pool of fragments that stop at all of the potential C's. So some of the times it's going to stop here. Sometimes it's going to stop at the next C, the next C, the next C, the next C. Each fragment is a little bit longer than the others. We can separate those fragments and identify them based on their size using electrophoresis, which is shown here. So here's a little actual fragment of an auto radiograph sequencing gel. Okay. The smaller fragments are going to be at the bottom, and the larger fragments are going to be towards the top. And you begin reading at the bottom, and you say, okay, what's the smallest fragment? What nucleotide stopped at the smallest fragment? Well, it was an A. So the very first nucleotide past the primer would have been an A. What's the next smallest? It also stops at an A. What's the next smallest? It stops at a T. What's the third smallest? It stops at a G. And so by reading this up like this, you're really reading the order of the bases where it stopped, each fragment length representing termination at that particular base. And if it was in the A stopped by A's, there was an A at that position. If it was stopped at the C's, there was a C at that position. So this allows you to read these sequences: A, T, G, C, G, C, T, G, C, A, T, C, G, T, A, G, C, T, A. That's how you determine the sequence of DNA. Now, this particular type of process is a little bit time consuming. You had to set up 4 separate reactions, you had to mix 4 separate things, and you had to use radioisotopically labeled nucleotides to be able to detect the exposure of the fragments to X ray film and then read that X ray film. So, I thought, well, let's speed this up a little bit. Instead of using radioactive isotopes, what if we attached a fluorescent dye to each different dideoxynucleotide so that we could track it by fluorescence? And we could do that using a spectrophotometer, and we would not need to separate them into 4 separate columns, but we could allow them to run through a single column and simply focus a laser beam so that it would determine what color was fluorescing, and by what color, what nucleotide was present that at the end of that particular length molecule. So this is how dideoxy sequencing is done today. And these machines can operate and sequence thousands of base pairs per minute. So what you're produce is a trace of intensity. So a fluorescence for A, fluorescence for A, fluorescence for T, fluorescence for G, fluorescence for C. And this is put through what's known as a capillary electrophoresis. So this would be an electrophoresis tube that's usually 4 to 8 feet long, about the diameter of a human hair. And the DNA molecules can flow through it, and it can separate everything from, say, 15 base pairs up to 5,000 base pairs in length. So with enough separation to determine what the last nucleotide was on each one. Here's an example of that type of analysis. All right, so we'll stop there and we'll talk about some other aspects of gene expression coming up next.